process of recombinant DNA technology. 1. Isolation of genetic material. The first step is the isolation of desired gene from a cell and the digestion of bacterial cell wall by enzymatic action. This is achieved by treating the bacterial cells of plant or animal tissue with enzymes like lysozyme bacteria, cellulase, plant cells, and chitinase, fungus. Genes are located on DNA molecules centrifugation with proteins such as histones. RNA can be removed by treatment with ribonuclease. Proteins can be removed by protease. Other molecules can be removed by appropriate treatments and further subjected to gradient centrifugation. Ultimately, purified DNA is precipitated out after the addition of chilled S. 2. Cutting of DNA at specific locations. The purified DNA is cut into number of fragments by enzyme called restriction endonucleases. Each restriction endonuclease functions by inspecting the length of DNA and recognizes a specific palindromic nucleotide sequence in that DNA. Then it will bind to the DNA and cut each of the two strands of the double helix at specific points. These are called molecular scissors. They were first discovered by Nathans in 1970. Over 250 restriction enzymes have been isolated so far. When linear DNA is treated with restriction enzyme, a large number of DNA fragments are formed. The resultant fragments are separated by gel electrophoresis. Finally, the desired DNA fragments are selected by Southern blotting technique. 3. Amplification of gene of interest using PCR. Once the desired fragments of DNA genes are obtained, they are inserted into suitable vector to produce indefinite number of copies of genes. This is known as gene cloning. A cloning vector acts as a vehicle to carry the desired gene. An ideal cloning vector should have following properties. 1. It must have low molecular weight. 2. It must have unique cleavage site for the activity of restriction enzymes at single point. 3. It must be able to replicate truly inside host cell after its introduction. 4. It must contain genes which provide resistance to antibiotics. Several types of vectors are used in recombinant DNA technology like plasmids, phages, etc. The enzyme DNA polymerase helps in replication. If the process of replication of DNA is repeated many times, the DNA segment can be amplified to approximately billion times. That is, 1 billion copies are made. Such repeated replication is achieved by use of thermostable DNA polymerase, which remains active during high temperature induced denaturation of double-stranded DNA. The amplified fragment of desired gene can be used to ligate with vector for further cloning. To isolate a plasmid, the bacterial cell is treated with EDTA, ethylene dimine tetraacetic acid, along with lysozyme enzyme to digest the cell wall. Then the bacterial cell subjected to centrifugation in sodium lauryl sulfate solution to separate the plasmid. The isolated plasmid DNA is cut with same restriction endonuclease enzyme. The enzymatic cleavage cuts circular plasmid into a linear molecule having sticky ends. The two sticky ends of this linear plasmid are joined to the ends of the desired gene. The enzyme DNA ligase joins the complementary ends of plasmid DNA with that of desired gene by covalent bonding to regenerate circular hybrid called recombinant rDNA or chimeric DNA. 4. Introduction of recombinant DNA into the host. The recombinant plasmid is transferred into a suitable bacterial host cell, generally E. coli, by a method known as transformation for the expression of desired gene. The cells into which the recombinant DNA are inserted are called transformed cells. Example, if rDNA bearing gene for resistance to an antibiotic, example ampicillin, is transferred into E. coli cells, the host cells become transformed into ampicillin-resistant cells. Only transformants will grow on agar plates containing ampicillin. The transformed recipient cells will die. The ampicillin resistance gene is called selectable marker. Bacterial cell walls are not ordinarily permeable to such recombinant vectors, but reaping in dilute solution of calcium chloride renders the bacterial cell wall permeable to the recombinant vectors. Inside the host cell, this rDNA starts replicating. The transformed cell will begin to grow on medium and divide as separate units. The replicate vectors in each cell are passed on to daughter cells, giving rise to clones.